Let's talk about the biology lab just for a minute or two. So labs start tomorrow, Thursdays, on campus. So labs start there at 2 o'clock and they're scheduled to 5 o'clock. Most of the labs do not go till 5 o'clock. Most of the labs are, I would say, 2.5 hours, but be prepared to be there the full 3 hours because some of the labs do last that long. It's in room CC 234. So if you don't know where that is, you may want to show up on campus early to locate the lab. So what are you going to need for the lab? You're going to need a lab manual. The winter 2022 edition, I think the cover is yellow this time around. Last time I checked, they were in the bookstore, and that was actually a couple of weeks ago now. So they should be in the bookstore. And if there's not one in the bookstore, ask, because they can print one off right away for you. So you do want to make sure that you have that available. I think everybody has submitted their safety training, so I'm not going to worry about that too much. And I will point out safety equipment and a few other things when we get to the lab. It's always important when you come in, you wash your hands. Always important when you go out, you wash your hands. And I'll do my best to try to remind you in the actual lab. One of the main things that you're going to need is a lab coat. So you can buy lab coats at the bookstore, and they must be worn at all times in the lab. And you're not to wear them in the hallway. So labs only. And that way, if you get anything on the lab coat, it's not going out into the hallway or anywhere else. And hopefully the lab coat is protecting you a little bit from anything in the lab. Just in general, another thing too is, I think I mentioned the footwear. Do make sure you have proper footwear, something reasonably comfortable. You're going to be on your feet for a little while, and it has to cover the whole foot. And there's some samples of some different types of footwear. And just a reminder that you do need to have your ankles covered. Anything down there below needs to be covered. So make sure you wear socks or pants that go all the way down, and you'll be in good shape. A couple other reminders. Contact lenses are not recommended for the labs. They're not allowed at all in the chemistry lab. In biology, I wouldn't recommend it. We do work with solvents sometimes, and solvents like to stick to plastics. And so it's not recommended that you wear them. Safety goggles for lab 3, you're going to want a pair, or at least sharing a pair with somebody. And I'll point out when it's necessary to wear gloves. So just a reminder of the stuff that you need for the lab manual, your lab coat, goggles for lab 3, a pen, a marker, and a ruler, and basically everything else leave behind. You can bring your phone, obviously, if you want, in your back pocket, but it shouldn't be out on the bench, is really why I have it on that other side. Anything else you can leave in your locker or in your car or in the hallway or wherever. We just can't have all this extra stuff in the lab. So just a reminder that the lab manual has some introductory pages. I'm not going to go through all of that stuff. But it's a good idea that you do review those first few pages. So when you get your lab manual, flip through there. There's information about evaluation, about absences. So if you have to be absent for some reason, you need to let me know. And I may request a doctor's note or something like that. I'm not really sure actually what our policies are this semester in terms of how we're monitoring that, because of course some people may have to isolate due to COVID. If you do miss a lab, you are going to still be responsible for the work. Meaning the lab reports and things like that. And so we'll just have to deal with that on a case-by-case basis. So more on the lab tomorrow. I'll give you a bit better of an overview when we're in the actual lab. So one other thing to keep note is that we do have some reports, and I'm going to start talking about report writing starting on Friday once we've had our first lab. And do always take a look at the lab schedule to make sure that you are paying attention to when things are due. And we'll talk about, like I said, the lab reports and how to do them as we go along throughout the semester. Last thing is, don't forget to clean up. Every lab we have cleanup protocols. Usually we have bins for waste and bins for beakers and bins for test tubes and things like that. And just make sure you put things in the right place. It certainly saves us a lot of time, and it's just nice courtesy for anywhere you're working. Always clean up after yourself. Okay, so let's get back into topic 2. And topic 2 last day, I had just thrown out some introductory chemistry and threw out a few terms. And today we're going to get right into and start and talk about the lipids. So let's talk about lipids. Lipids, there's a whole bunch of names. Some people call them fats, and there's different types of lipids. And probably the most common terms that you've heard for lipids are actually these terms here, saturated and unsaturated. And you take a look at these. I thought I had, I'm just going to skip ahead for a second here. I thought maybe I missed a slide. There we go. I feel like there's a slide missing. Hold on a second. I am just going to get out of my PowerPoint. Oh, I see. Okay. Sorry about that. I'm just getting a little bit ahead of myself in terms of where I thought the slides were, where they were. I'll put that back up. So saturated and unsaturated. I'll show you some chemical structures in a minute. Saturated fats, generally, they pack really well. So you can see they kind of look like zigzaggy molecules. And if you remember your basic chemistry, you've got those London dispersion forces. And when things pack really well, they can kind of get close together and have decent London dispersion forces. Unsaturated fats are not, they just don't stack quite as nicely. And I'll show you why in a moment about some chemical structures. But in terms of what we usually call these things from a generic stance or from a dietary stance is often things that are saturated we call fats, and things that are unsaturated we call oils. And that's because fats are solid at room temperature and oils are liquid at room temperature. So I'll show you some pictures here of some fats and fatty acids. So in this regard, we're actually talking about fatty acids, by the way. And so I really feel like I'm missing a slide or two. Ah, yes, I am. Okay. Hold on a second. Let me just go back to a slide here. I did mess up. Totally one slide ahead of where I meant to be. I want to talk about the lab, and I inserted that. And there we go. Okay, that's the slide I thought we were looking at. I really apologize for that. All this online stuff is really, the organization is just a little bit of a different deal when you're dealing with online here. So these saturated, unsaturated fats, we're talking about fatty acids is really what we're talking about. There's the chemical structures, and I want to show you the difference between the saturated and the unsaturated. In terms of the chemical structure, they're called a fatty acid because they actually have an acid. And this particular functional group right here, this is called a carboxylic acid. Carboxylic acid. If you take some more chemistry courses, you're going to see a lot of carboxylic acids. The rest of the molecule, you can see this part here, is completely hydrogen and carbon. So that's kind of one of the #1 things to look for when you're trying to recognize a fat or a lipid of some sort, is that it's almost completely carbon and hydrogen. And carbon and hydrogen have basically the same electronegativity. So when they form covalent bonds with each other, they are forming non-polar covalent bonds. So this whole section here, we would call non-polar. And another term we might use for that is hydrophobic. And hydrophobic, of course, means it does not dissolve in water. And so what you have is this large molecule, most of it is hydrophobic and nonpolar, and that becomes the kind of main characteristic of this molecule. And so it's hydrophobic. And so that's actually a definition of lipids, is that they are biological molecules that are not soluble in water. The most common type are these fatty acids. And you can see on the left, this one, the one on the left is saturated. And so what's the difference between saturated and unsaturated? Unsaturated have these double bonds. And these double bonds kind of make the molecule, you can see, rather than a perfect zigzag, it has kind of kinks in it. And that was what this slide here was showing you in terms of how they pack or don't pack together so nicely. And the consequence is that the saturated ones are usually fats, and they're solid at room temperature, and the unsaturated ones are usually liquid and called oils at room temperature. So this is not necessarily how we find most fats in nature. Most fat molecules are actually TAGs. So what's a TAG? TAG is triacyl glyceride. So there's going to be 3 of something. In fact, we're going to have 3 fatty acids, and I'll show you the other 2 here in a moment. So this here is the fatty acid, this orange molecule here. You can see it's the same structure I just showed you on the previous slide. It's got a bunch of hydrogens and carbons, and it's got that little carboxylic acid there on the left-hand side. And the glyceride actually stands for glycerol, which is this thing here. And glycerol, you can see, is sort of like a fat molecule. It's got some alcohols on there. It more closely resembles a carbohydrate technically, and we'll talk about carbohydrates a little bit later. But basically, you have 1 glycerol molecule, and you get 3 of these fat molecules. If you take a look, what's happening right here is this condensation or dehydration synthesis reaction. So you're taking away a water molecule and you're joining those 2 things. So a TAG, or a triacylglycerol, is actually 3 of these things all joined together, and it kind of looks like that. So before we get any further, I want to stop and show you what I have going on here. Normally I would do this on the whiteboard in class, and as I'm talking, I make some notes as we go along. So I want to start making some notes here for the lipids, and we'll come back to carbohydrates and proteins later, and I'll come back to lipids as well, because I'll have some more information to fill in. So you can see I've made a table here. We've got the type of macromolecule, we've got the basic unit, a little bit about the chemistry, what are the linkages and what are the functions. So let me fill in some of that information. And if you miss something, don't worry, I'll come back to this repeatedly as I'm talking and going through the notes. So the lipids, the basic units, remember we had fatty acids and glycerol. And so that's one way to think about these things. And like I said, I'll fill in some more things here in a minute. So what about the chemistry? Chemistry, we can say, are mostly carbon and hydrogen. So we'll say non-polar hydrophobic. You are going to see other atoms in there, but in terms of the majority of them are carbon and hydrogen. Linkages aren't as important for this one, but if you'll take a look back at the slide, the linkage, at least in triacylglycerols, is called an ester linkage. And if you don't know your chemistry well enough to know what an ester linkage is, I don't want you to worry about that one too much. And then, of course, the last thing is, what are we doing with lipids, right? What's the whole function of these things? You probably know that fat is part of our diet. So we can call it energy, and it's an energy source. And of course, if you eat too much, it becomes a form of energy storage. So I'll put that down as well. So that's a good start. I will come back and fill in a few more things for lipids, but I'm going to go back to the PowerPoint now. So there's our TAG, or our triacylglycerol, and that is also called a fat molecule. And that's usually the biological form. So when we store fat in our body, that's how it's stored usually. When you eat food with fat in it, whether it be vegetable fat or oils off of an animal source, it's usually some sort of triacylglycerol is the main form of fat molecules. There's a little diagram there, or not a diagram, sorry. This is actually a colorized micrograph, and you probably know that fat is, of course, hydrophobic. So hydrophobic. And what does that mean? It means it does not dissolve in water. Technically, hydro means water, and phobic means fearing. And I think that's from Greek. I'm not entirely sure what the origin of those languages are. For hydrophobic, that's what it means. And so what you're seeing in this diagram actually is some fat in some human fat tissue, so human adipose tissue. And the fat molecules, they don't dissolve with the water, and they turn into these little kind of blobs or spheres or whatever you want to call them. And you can see they're labeled there as fat droplets. They're not dissolving with the water. So I want to talk about some other lipid molecules that are important, at least for this course. Another type of important lipid molecule is something called a phospholipid. So let's take a look at the structure and see what it is. If you take a look here right in the middle, we have glycerol all over again. And we have a fatty acid and another fatty acid. So what we have is basically a tag, a triacylglycerol, but that third fatty acid has been replaced by this group here. So that group has a phosphate in it, and it has a choline. Now there's different types of polar groups you can have on there, and they usually have some sort of phosphate in them. So we call this molecule a phospholipid. So this is kind of a unique type of molecule because it has charges in that polar section. And in fact, you can see they have these labels here. They call this the head, the hydrophilic head. Hydrophilic means it's water loving. It can dissolve with water. And we have these hydrophobic tails. So now we have a molecule that's kind of 50-50 in terms of its chemical nature. Half of it is water fearing, half of it is water loving, half polar, half non-polar. So this is kind of a unique molecule. One thing to note, and you probably know a little bit about phospholipids, learning from them in high school, is that they're part of membranes, and I'll talk about membranes in a minute here. But you can see what they're showing you as a structural formula, and it kind of gets tedious to draw that out every time. And so usually what the textbooks do is they draw the phospholipids looking something like this. You can see it has a polar head and these hydrophobic tails. So if you are to take some phospholipids and throw them into liquid, this is what you get. And so you can see some of them are forming blobs. There are different types of phospholipids, but the majority of the phospholipids are actually forming something that is a little bit different. And it's not these spheres anymore because there's more of the molecule can actually interact with the water. And what we actually get are many of the phospholipids form what is called a bilayer. And you can see that shown here in this lower diagram. The spheres are called micelles, by the way. And so what we have is this bilayer here with these little polar heads. That's a polar head. And this is the non-polar fatty acid tails. And so the polar heads are interacting with the water, and the tails are interacting with themselves. And so now we have a barrier, a layer, and this is actually the main structural component of your cellular membranes. So here's our little cell. You can see our cell is made of water. We've thrown in some lipids, and so this is part of a huge structural component of cells. Some cells have, of course, membrane-bound compartments as well. So let's go back to the Word document here, the whiteboard, and we'll fill in some more details here. So maybe I'll put this in its own section. We have phospholipids. So maybe if you really want to expand, you could put in triacylglycerides are 3 fatty acids and 1 glycerol if you want. Okay, and then over here, I'm going to put membranes. All right, so there are a few other fats that are worth mentioning. And so I will come back to this in a moment here. So there's our little cell. Like I said, a big part of fats are actually cellular membranes. Huge part of whether you're an animal or a plant or a fungi, much of your fats, your lipids are actually part of phospholipids. If you're an animal, that can be a big part of your energy storage as well, and depending on your energy intake and your caloric intake and things like that. So there are many types of lipids, by the way. Pretty much anything that fits in that category of being water insoluble, made out of carbon and hydrogen, is a lipid. And so I'll show you a couple other types of lipids. One is this other group called the steroids or the sterols. And in terms of their chemical structure, what we're looking at is these ringed compounds. And so if you take a look, it's a 4-ring structure. And there's an example there, which is testosterone. Testosterone is a hormone, of course, and it helps building muscle mass and also contributes to things like body hair and whatnot. And so it turns out, if you know your chemistry, each of these joints right here in the molecule actually represents a carbon and a couple of hydrogens. So I could draw these in. And so it turns out that this molecule here is just a whole bunch of carbons and hydrogens. It's loaded with carbons and hydrogens. So that makes it a lipid. A couple of oxygens, not a big deal, not enough to really make it polar, 2 oxygens amongst all those carbons and hydrogens. And so it makes it a lipid. I know it looks nothing like the other lipids, and that's the one thing about lipids is a lot of them don't look like each other because they're just in this group of molecules that are kind of generally grouped as being insoluble in water. There are other steroids out there. I'll show you some other ones that you may or may not be familiar with. You may have heard of cholesterol. Cholesterol, of course, is in animal tissues, and we talk about it in our diet, but it's actually really important for your membranes. It has a lot to do with altering and dealing with your membrane fluidity. So we'll come back and we'll talk about membranes in topic 4, and we'll talk a little bit more about cholesterol. You may have heard of some of these hormones here. I mentioned testosterone. There's also estrogen, cortisol, all sorts of different signaling molecules. That last one, solanidine, I think, I'm trying to remember what it is. I think it might be an insect pheromone. So a different type of biological function. There's a whole bunch of them out there. They all have that 4-ring structure. So kind of the main takeaway is a steroid is a molecule with 4 rings, could have a variety of different biological functions. Okay, so this is my reminder to make a table of these macromolecules. And I'm going to go back to that table and I'm going to fill in the steroids. So here we go. So steroids. And steroids are involved in membrane structure as well. And we also have hormones, and I'll just put etc. And if you're curious, you can look up some of those other steroids anytime you want. But mainly in this class, we're going to be talking about cholesterol. Okay, so just a reminder that lipids, in terms of the chemistry, are mostly carbon and hydrogen, and they're hydrophobic molecules. Phospholipids are sort of in a funny group where they're kind of half hydrophobic, half polar, and we'll talk about that a little bit more when we do talk about membranes, because there's actually a special word for that. The word is amphipathic, but we'll get to that as well when we talk about membranes. Okay, so back to the PowerPoint. Like I said, if you don't get all that, you can always come back and to either the video, or we'll come back to it when we talk about carbohydrates. So let's talk about carbohydrates now, and we'll see what some of the differences are between carbohydrates and fats. Carbohydrates are also known as sugars or saccharides, and all those words are used somewhat interchangeably. Carbohydrates are famous, sugars at least, are famous for being sweet and tasting delicious. But in terms of the chemistry, what we're looking at is a bunch of carbon and hydrogen, but also a bunch of oxygens. And these oxygens form alcohol groups. So if you ever see a biological molecule and it has a whole bunch of alcohols on it, usually there's almost 1 alcohol group per carbon. So maybe you have 6 carbons and 5 alcohol groups, or even 4. And that is a carbohydrate. So if you take a look on the right-hand side, this here is sucrose, right here. And sucrose, you can see it has a whole bunch of alcohol groups on it there. I've got a couple of them labeled there in the diagram. Sometimes alcohols have these other groups here. They have carbonyls. We don't actually see any carbonyls on this particular sugar. But what you do see are some esters, and esters are basically a carbon linked to an oxygen linked to a carbon. So there's another ester here right in the middle. But the main one to look for is a whole bunch of alcohol groups. The other thing to look for is that a lot of carbohydrates are found in these ring structures, You know, so if you see a molecule and you've got a hexagon or a pentagon, usually with some sort of water somewhere on it, or not water, I mean oxygen on it, that means you've probably got some sort of carbohydrate. Let's take a look at some of the carbohydrates. I mentioned that they're usually found as ring structures, but not always. And so this part of the textbook is an image from the textbook, and it's showing what a bunch of them look like when they're in the linear form. So they do exist in both forms. But the main thing to look for is look at all these carbohydrates, or look at all these alcohols. They're all over the place. There's some carbonyls. And so that tells me right away that these are carbohydrates. So the individual carbohydrates are called monosaccharides. Mono means one, and saccharide means sugar, of course. And these have 3 to 7 carbons. The most famous ones are the ones with 6 carbons, and the most famous carbohydrate of all is probably glucose, followed by some of the other ones, fructose or sucrose or whatever. There's the alcohol. This I animated to show some labels on there, and the carbonyl as well. So I wanted to show you this because these carbohydrates can exist in both forms. The textbooks, you know, they show these linear forms often because it's convenient. They're much easier to draw. But biologically, in an aqueous solution, about 87% of the time, the equilibrium is over here in the ring form. So that's how you're going to see them most of the time in textbooks anywhere. You're going to see them in this ring form, even though it's more convenient to draw them in the other form. A huge percentage of the time they're found in the ring structure. Sometimes we see different types of abbreviations around these ring structures. You can see, I don't know why they're calling that the abbreviated ring structure. I mean, there's not much difference. I guess you're not seeing the carbons at the joints, right? Is the difference what you're seeing in that diagram? Sometimes they're even simplified even further. So if you can have a monosaccharide, then you must be able to link them together and make longer saccharides. And so if you take a look at this here, this is glucose and fructose. We can link them together. And here is our dehydration reaction again, dehydration synthesis. And we are making a disaccharide, right? Di means 2. So over there on the right is sucrose. That is common table sugar. And there it is. And this linkage here you have on a carbohydrate is called the glycosidic linkage. It's actually just an ester linkage, but when it's in a carbohydrate, we give it a fancy name. So now seems like a good time to maybe fill in some of this information in the Word document and give you some information there. So for carbohydrates, the basic units are called monosaccharides. Examples might be glucose, fructose, ribose, and there are many others. So the chemistry, they have carbon, hydrogen, and oxygen. Sometimes they have other elements, and I'll show you some carbohydrates that do. So maybe I can put that in bracket and we'll say sometimes nitrogen. The other thing, remember, is that they usually have many alcohol groups. Okay, there we go, groups. And alcohol groups are basically OHs. And it looks like my document has reverted to French for some reason. And so now the spelling is all incorrect for some reason. Let's see if I can fix that really quickly here with Canadian English. What were the linkages called? They were called a glycosidic linkage. So anytime you see that, that glyco thing, so right here, glyco. Glyco means carbohydrate. It doesn't mean glucose, it means carbohydrate. So anytime you see glyco, it means you've got a carbohydrate of some sort. So what are carbohydrates used for? We can We obviously know they're used for food, so we can say they're used for as an energy source. And they are also used as energy storage, and I'll give you a few examples in a minute. So maybe I'll put that as its own group, energy storage, and I'll show you the examples of that in a moment. But mostly we're talking about starch and glycogen. So starch is in plants and glycogen is in animals. So I'll come back to this. We're not done talking about carbohydrates, so I'll return to this in a second. So there's sucrose there. Sucrose, of course, is a food source. Humans don't store sucrose, but some plants do. It might be stored in the fruit. A lot of berries and things like apples have sucrose that makes them taste nice and sweet. And we can go further. We can have trisaccharides where we're linking 3 together, or you can have polysaccharides where you're linking many together. And I'm going to kind of come back to this in a bit. This is just a little bit more chemistry for you. You can kind of link them in different ways. And that's what this diagram here is showing you. There's 2 different ways that we can link up polysaccharides. It's a lot more complex than this and not really worth getting into for this class. But if you take a look at on the left, I have starch. And you can see the starch that each of these glucoses is oriented in the same direction. On the right, I have cellulose. Cellulose, we have this one is oriented upside right. This one is upside down, upside right, upside down. So what does that do? You would think it would not make a big difference, but it actually does. It has a huge difference in terms of hydrogen bonding and actually makes cellulose a much stronger structure. I'll come back to cellulose in a minute. Let's talk about starch and glycogen. So starch, as I mentioned, is found in plants, and this is how they store their food. So they make food by photosynthesis, and they might make some glucose molecules, and then they link up all these glucose molecules, and you get starch. People might talk about different kinds of starch. You can see on the right there, there's amylose and amylopectin. Amylose is just the glucose linked up one by one. The amylopectin might have some branches in it. And this is a big part of our food and a big part of our diet. Starchy foods include things like potatoes and carrots and rice. All those things are loaded with starch, and it's a good source of energy. Animals make a very similar molecule called glycogen. And glycogen, if you take a look at the structure shown on the right, it is very similar to starch. It just has many more branches. We store glycogen in our muscles, and we also store it in our liver. And those are the only 2 places we store glycogen. Glycogen is really important because glucose is the energy that we need for our brain. In fact, glucose is the only energy our brain will use. And why is it branched? Because when it's branched, we can just grab these glucoses. There's a whole bunch available at the ends really easily. It's much easier to grab them and get a whole bunch of glucose quickly. But if you need to go for a jog right now, or if you need to do some vigorous exercise of some sort, or your brain just needs it now, you can get it now. Whereas starch, if there's only 2 ends on a molecule, you can't get a lot of glucoses very quickly. Okay, so let's talk about cellulose. Let me go back to that slide here in a moment. This one here, I was showing you that the linkages between starch and cellulose, there's something different about them. It has to do with the orientation of each of these glucose molecules. Now these structures here that we're seeing, these hexagonal structures of glucose, they're kind of just a flat version of a 3D molecule. And so this other slide that I popped up here, this is showing what glucose looks like a little bit more in 3 dimensions. And one of the things it's showing here in this diagram as well is all these dotted lines here. So right here, and here, and here, and here, and here, and so on. Those are all hydrogen bonds. And hydrogen bonds are weak molecular forces. They're basically dipole-dipole interactions. But when you have thousands of these things, they make the structure a lot more rigid and much tougher than starch. Starch will dissolve easily, even in your mouth. If you chew a cracker for long enough, it starts to, you can break it down in your mouth. Cellulose, on the other hand, is not digestible by humans. And has a very different function as a carbohydrate. So you can see right up at the top, it says it's used for the cell wall in plants. And here's the diagram from the textbook. You can see they're trying to show the same thing, trying to show all these hydrogen bonds in there. You can see those dotted lines are just a little bit on the small end. They form these fibers called microfibrils, and they form up the plant cell wall. So a cell wall is much tougher than a membrane. It's much tougher than starch. It's just like the wall of your house. It's very, very tough material. And so it's good material to protect the plant cell. I'll come back to the Word document in a minute and fill in some of these details. This is just, I guess I'd said it already, cellulose is so tough that we can't even digest it. So a lot of the plants we eat, parts of the plants are cellulose. If you eat celery, there's a lot of cellulose in whole grain foods, and we actually call that stuff, that cellulose, we call it fiber. And it's actually good for your gut. It helps clean your gut, and it helps attract the correct bacteria, but we don't actually digest it as food. Some animals do digest cellulose as food. Cows and a lot of herbivores eat plants all the time, and they digest cellulose with the help of bacteria. They can't do it on their own. So before I get to the notes, I wanted to show you a few other tough structural polysaccharides, because we are going to talk about some of these structures. The next one that's worth talking about is called chitin. So chitin is very similar to cellulose, except for the monomer is something else. The monomer is called N-acetylglucosamine, which can be abbreviated as NAG. And it's very, very similar to glucose, except for it has this group on here. This is called an N-acetyl group. So kind of like glucose, but it's decorated with a couple of extra functional groups. Same kind of structure as cellulose. It's got lots of hydrogen bonds, and this forms the fungal cell wall. So fungal cell wall. You can see it's mentioning the exoskeletons of insects and other arthropods, and it's tough enough that it can actually be used as surgical thread, and which it is sometimes. There's different types of surgical thread, but it's a good structure for that. Another polysaccharide we're going to be talking about in topic 5 is something called peptidoglycan. So take a look. There's that glyc again. This is a carbohydrate, but this one also has a peptide attached to it. So there's the monomer of peptidoglycan. We've got some NAG, that's N-acetylglucosamine, and NAM, N-acetylmuramic acid. I'm not going to expect you to know NAG and NAM, by the way. And you can see they're just like glucose, but they're even more decorated. In fact, NAM has this little peptide part on it. So it's peptidoglycan is describing the molecule. It has peptides and carbohydrates on it. So these kind of link up in this big mesh, and lots of covalent bonds, we call cross-links, are binding this molecule together, and it's forming the bacterial cell wall. So bacteria, fungi, and plants all have cell walls, and they're made of very tough material. So what I'm going to do is I'll add that to the Word document. All right, carbohydrates, functions. Here we go. So structural, I'll say, and we've got plant, fungi, and bacterial cell walls. And that would be cellulose, chitin, and peptidoglycan. There's one other function of carbohydrates that I have not added to the PowerPoint slides, but I'm going to add to this this document now. Is we're going to call them I'll call them glycolipids, and they're on membranes. And we're going to talk about those in topic 4. So it's just worth throwing that into this document at the moment. All right, so there is our peptidoglycan. There's our little cell. We've got our lipids in there, the phospholipids making the membranes and making some compartments. And let's give it something to eat. So there's some food, could be some starch, could be some glycogen, depends on whether it's a plant or an animal. And it's got a cell wall. So I guess this one must be a plant or a fungus. And the cell wall, of course, is a lot more structural, not easily digestible. Okay, I have a Kahoot for you. So load up the app and we will do a Kahoot.